that. And now I have, let's move on to part 5, which is going to be shorter than part 4 by quite a bit. But it's going to be the questions related to antibodies, because they're talking about now making a bunch of antibodies. And remember that good antibody versus bad antibody. If you're running an ELISA, you want a single sharp band associated with the protein you're looking for. You don't want it just binding anything. That's if you're running an ELISA. If you're the human body and defending yourself against infection, on the other hand, non-specific antibodies might actually be a good thing. It could be a good thing to accidentally have an antibody that accidentally binds this other virus that I'd never seen before. That is called cross-reactivity. It's the question of, can an antibody be, how non-specific can an antibody be? Can we have an antibody against one virus that also binds another virus? And so the vocabulary words that I want you to use from this are epitope and paratope, actually more epitope than anything else. Here's a B cell at the very bottom, and you see the B cell has a B cell receptor, looks exactly like an antibody, right? And it's binding an antigen. The antigen contacts the antibody through its epitope. The epitope is a sticky part of the antigen, and the antibody part that binds it is called the paratope. Usually I just call it the antibody, honestly, but epitope is a very useful word. So our question is, is there always a one-to-one relationship of these? Can one paratope bind different epitopes? Or can one epitope bind different paratopes? The direction of the question that's really interesting is, can we have one antibody that can do double duty for us? Can we have antibodies that moonlight, in a sense, like do two jobs for us? Okay, so that's what the epitope versus paratope thing is. Now, I'm going to start with the chemical example, because there are clearly cases where antibodies can bind more than one thing. And this can cause a problem. In fact, this is a problem for people who are using it for a blot. Here is what they have. They had a monoclonal antibody, so a single sequence, single structure of antibody. It was selected for specificity, and here's RAS again, by the way. I did not plan this, but I just realized that they're studying RAS like probably half the biotech industry. They're bound to the mouse version of RAS. And it was a peptide that was a fragment of the mouse version of RAS. So that's how it was raised. They put the mouse RAS peptide into whatever organism made the antibodies, and it made antibodies against it. They stuck, great. But then they found out there's a problem. It also binds recombinant full-length mRAS. Well, that's not too much of a surprise. It binds the full-length version as well as the fragment. But it also binds HRAS, which is rather different than RAS. Right here, they have squares around all the identical things, and you see it's about 50% identical. And in fact, for the peptide region, the peptide that they raise it against is way down here. And there's only like, what is this, 5 boxes that are identical? There's some similarities, but for some reason, this peptide raised against this sequence will also bind this other sequence, and they don't want it to, you know? So this is a case where they don't want it to do things, and this is the evidence showing, basically with all of these, higher numbers means more binding, and so you can see that right here, you have the immunized rabbit, and you have plate-bound mRAS peptide, plate-bound HRAS peptide. When you take the immunized rabbit antibodies, the antibodies stick nicely to mRAS because they've seen it before. If you take them to the HRAS peptide, they don't stick very much. There's a little bit of binding when you get to high levels, but this is nice and specific. Here's where they did ELISA with a monoclonal antibody rather than serum, and you see that's even better. You get no binding to the human peptide. So this looks great. The problem is when they started to use full length. Then they did ELISAs where they had purified rabbit monoclonals against plate-bound recombinant full protein or recombinant full human protein. and they saw the human protein was binding almost as much as the mouse protein. And then they're like, oh man, you know, that's annoying. So then what they did is they put the antibody on the plate, and they did it backwards. They wanted to see maybe there's some problem with the antibody non-specifically sticking to the side of the plate. So let's reverse it so that the problem won't happen anymore. Well, then again, they find the less binding. That makes sense because you have 2 binding sites rather than 1. But also you have the same pattern with human and mouse. So it's like, okay, it works great for the peptides, but there's something about it that's also binding full-length human. So they wanted to be scientists about it, and they wanted to look and see what it was. They did a bunch of investigations with different, they basically made a bunch of peptide fragments, and they saw which ones bound, and when you had the whole peptide fragment bound, they said, okay, these peptide fragments always bind. You know, they did this back and forth, and they figured out which things always bind. And you notice that here's the MRAS fragments. There's a lot of things that actually bind there, but when you look down here, you see a lot of binding at the end where the peptide is. And then for the human, you also see a lot of binding at the end. And when they looked at this pattern, it was always centered around the positively charged residue. So look at this sequence. It's the same sequence right here and right here. I think you can see this one. Okay, so positively charged residue seems to be what this antibody likes to bind. Remember Delano and Wells? They were like, well, maybe it's non-specific, it's binding so many things because it's always binding just positively charged residues. They found out that wasn't true in that case, but it looks like it's true in this case. So look at this and tell me, are there any things that are non-identical residues, and yet they might also be both positively charged? They might allow the antibody to bind it because it likes positive charges. Do you see something where it's different? So here you have 2 lysines, so those are the same. But do you see something that's different, but it's chemically the same and positively charged? Do you see it? You have a KR there. And so because there's so many Ks here, they think that that this K-rich sequence, the antibody just likes to bind this. This is the center of the epitope. And because even though the rest of the peptide is very different, this part is very much the same. The antibody cares about this part more than anything else, and so it sticks to both of these. That KR right there, to me, that seals the deal. Also, the fact that these are all concentrated on this side of the peptide. So yeah, this looks like it's a case where the positive charges rule the day, and they explain what this one peptide just likes positive charges in that arrangement. But of course, this is not just an academic question. SARS-CoV-2 is a coronavirus, and it is not the first coronavirus your body ever saw. There are circulating common cold viruses that are also coronaviruses. And in fact, if there's SARS-CoV-2, you also know there must have been a SARS-CoV-1. The good news about CoV-1 is it never really got out of the region. It stayed in East Asia. They were able to keep it from spreading. But I remember when it happened, I never thought it would have a progenitor that would be an even worse one. But here we are. And you see, this is a phylogenetic tree. SARS-CoV-2 and SARS-CoV-1 are pretty darn close. You also have MERS. Now, you've heard of MERS, is that right? Have you heard of MERS? There was a brief scare at the World Cup where some people on Twitter were saying that there was a MERS outbreak starting. MERS appears to be a more serious disease, but we see it less, so we can't be sure. But it appears to be more serious. It's a respiratory syndrome. It's a coronavirus. And it also has not spread, although it hangs out in camels. And so in the Middle East, when they had the World Cup in the Middle East, people would start freaking out. And they thought on Twitter they had evidence that a MERS epidemic was starting. I think it's worth it to say that people freaking out on Twitter is not always the most reliable way to get your news. But I was a little bit worried for a while because, you know, it could happen. I didn't think they had any evidence, and it ended up being that take was right. But down here, so you've probably never seen MERS, you've probably never seen SARS-1. You've probably seen SARS-CoV-2. I'm not going to ask for your medical history. I'm just assuming that most people have had it by now. Down here we have common colds, which you probably had at least one of these as well. See that there's 4 different kinds. HKU and OC43 are more similar. They're called the beta coronaviruses, whereas these ones are called the alpha coronaviruses. But actually, this fact gave me a little bit of hope, especially when people were freaking out about, justifiably early on, about SARS. But then it's like, how do you know how to trust? How bad is this going to be? And it's a novel virus, sure, but it's a novel class. It's not a novel class of virus, because we've seen beta coronaviruses before. So I wondered, could there be an antibody that binds to this and also binds to that? By the way, probably not. But there probably can be a T-cell response. We'll have to talk about that response on Friday, okay? But there is some limited antibody cross-reactivity. It's just not that you could be infected with this and have antibodies for that. They're too distant removed. But they found that some people with SARS actually had antibodies against MERS, which we know that there's no way they could have ever seen it, which means they must be cross-reactive. You must have made antibodies that hit a part of the protein that is similar enough for it to bind both proteins. So that's the cool thing. Now remember, I think I mentioned this briefly in a previous one, but remember that we actually had a sequence for SARS-CoV-1 and SARS-CoV-2. And you can see how similar they are. Here they highlight the differences. So all the blank spots are similar. And in fact, all the blank spots are identical. And then the similars are blue. So SARS-CoV-1 is very similar to CoV-2. And most antibodies, well, I'm not sure about most, but a significant number of antibodies that we developed against SARS-CoV-1 can also be used for SARS-CoV-2. So this came out very early on because somebody just went to their freezer where they had been working on CoV-1, they pulled out the antibody, and they found out it bound CoV-2. And it makes sense if you look at how similar they are. Here is a blot, by the way, that shows that there is binding right here. This is nice. These are showing different things, but these are decent antibodies. And you see that even these published data, you do have little extra bands once in a while from like aggregated protein or something like that. You are a little bit embarrassed, but this is still okay. But this is a SARS-CoV-1 and CoV-2. And this is using a polyclonal anti-SARS-1 antibody. So that before was a monoclonal, and now we have polyclonal from serum, and that's strong enough to bind. So this means that at least between SARS-1 and SARS-2, you have cross-reactivity. How far can you take it? And that's the big question to what's going on. So there's been several studies that have looked at that. Here's an example of antibody binding epitopes. And you see right here they have these things highlighted in red. These are the places where antibodies bind. And they found out that this is, these are the antibodies just in general to the individual things. But right here, they found that these antibodies bound, if you have an antibody that binds this sequence of SARS-CoV-2, then it will also bind NL63 at the same point, which makes sense because look at those sequences. They're only different at a couple places. And you can ask the question of how far does it go? Here, SARS-CoV-2 cross-reacts with this OC4 peptide, but it doesn't cross-react with the other 3 common colds. But the thing is, you can look at protein sequences, and you actually have a bit of a power with this because you can look at something and you can say, is it reasonable that these are similar, or which ones are the most similar? You can probably look at this one and say, just by looking at it, oh, that one looks more different than the other ones. So if these other ones don't cross-react, probably this one won't either. You can think like an antibody. And that comes from knowing your amino acids. I think that's kind of cool. So if your eye says they look similar, then they very well might act in similar ways. And so I could see these patterns, and I could say, okay, there's probably some cases where we have some kind of immunity to part of coronaviruses, but it's not going to be majority. And I think that has actually held up From common cold circulating, you have at most like 1/4 to 1/3 of full immunity against SARS-CoV-2. So it's definitely not something that's going to replace a vaccine, which will give you like 95% immunity. But it's something, and it might keep people, probably not from being sick, but it might keep them from going into the hospital because they had a common cold 10 years ago. But you'll never know that, unless we do a study on it. So here's another tree of the beta coronaviruses up here, alpha coronaviruses. And they studied them with DNA microarrays and bacteriophage display. But the whole thing is they basically found out in COVID-19 patients, of course they had immune systems, the antibodies that bound CoV-2. But look at all this blue they have. They bound CoV-1, they bound not MERS quite as well. You see how MERS is kind of light? But then you have these bat coronaviruses. These, by the way, are pretty close. But it's a bat coronavirus that's related to the HKU thing, and they found some antibody cross-reactivity there. See, the blue color is lighter. And also, by the way, this is not coming from just natural immunity to these viruses. If you take people who've never had COVID, they don't have any immunity to any of this. So this is starting to talk about the limits of our immunity. So there's another couple of studies that looked at how antibodies work and how many antibodies do we have. Like, have you ever wondered how many antibodies, different antibodies do I have against SARS-CoV-2 if you've been infected? We can actually answer that question if we have a lot of time and grad students in a sequencer. But here's one way that people looked at antibodies. They went to look for antibodies that a lot of people would have, not like some weird antibody that I have because I happen to have a fluke of the joining of the chains and whatever like that. But one that sort of comes from the genes, comes from the genes directly without many mutations. And that means that a lot of people would have it. So they screened a lot of people. They tried to find out antibodies that multiple donors would have in common. And so they call those public antibodies. The public antibodies that found epitopes that were prevalent. They were also looking for epitopes that weren't on the receptor binding domain, which is where most neutralizing antibodies are targeted, but it's also where the most mutation happens. So they went to look for antibodies that went outside the norm, and yet would also bind the spike at the N-terminal domain. And that's what they found. They found that there were like 4 donors that had antibodies, and these are the different antibody sequences. So down here you can see, you're not supposed to see the letters, but just look at the colors. Each of these is a different donor's antibody that binds SARS-CoV-2 in the same place. And so each of these comes from different people. And here at the top is sort of the germline antibody. It's the lineage antibody. The antibody that's encoded, and like people tend to make it over and over in their systems. And look how similar those are. So these people really are making essentially the same antibody over and over again. Different people making the same antibody. Maybe they're onto something, and maybe we can use this data to make more of this antibody for people. So of course they got a structure of where the antibody binds. Look up there, here's the spike. By the way, receptor binding domain is like here. And so you see that this is binding aside from the receptor binding domain. And you have the heavy chain and light chain of the antibody. You just have the fragment of it binding. So let's zoom in. Here's the antibody. Here's the N-terminal domain on the bottom. And look over here. You see, if you want to say which is the most important thing, lysine 147. That looks really important to binding. Also, the antibody fits into this groove between the N3 and N5 loops. The N3 loop is what contains lysine 147, and the N5 back here is arginine 246. Okay? So you don't have to remember those numbers because I did, and they're going to be important on a later slide. In fact, they're going to be important in this study. Totally separate study, same idea, but a different approach. What they did is they had 3 donors who had COVID-19, and they took their antibodies, and of course they made antibodies to SARS-2 spike. But look at this, because their
cryo-electron microscopy will actually pick up all of the antibodies. So spike, as you know, has this little triangle shape. Whenever you see these colored things, each of these colored things is a different antibody. So this is one person, and look at all the different antibodies they're making to spike. Their immune system really wants to get rid of it. And they seem to really like making antibodies to that little corner of spike over there. By the way, the yellow is receptor binding domain. The red is the N-terminal domain. So you have this other donor, same story. Another donor, well, they only have one, but it's in the same area. It looks like this area is something that these people like to make antibodies to. Well, let's zoom in. Here's all the different antibodies. There's antibody number 11 to antibody 15. And they're binding right here. You can see this just more resolution to the whole thing. And this is where it is on the SARS. And if you look there, remember the lysine that we saw? That's where it is. And so the other study, using different techniques, found other antibodies that like to bind this lysine and that like to bind the whatever the arginine or whatever it was over here. They like to bind these two loops. So this is a really nice confirmation. I love it when you can have different techniques to find the same thing. This is a really immunogenic part of SARS-CoV-2. And also I want to look at these residues. How much do they mutate in different variants of SARS? Is there pressure on them? And maybe if there's not, then maybe we can use that fact. So maybe this could be an Achilles heel for SARS, where it's a part that's easy for antibodies to bind. And by the way, think of this in terms of Delano and Wells. Remember accessibility and exposure, accessibility and hydrophobicity. If you're walking the line between those two things. Oh, and I forgot to bring your quizzes back. I've graded your quizzes. You did fine. I just forgot to bring them today. So sorry about that, but come to class on Friday. I'll bring your quizzes, okay? But mostly, I think several people answered that question, the accessibility versus polarity. It looks like that same trend is taking place here. This might be a bindable patch. Wouldn't that be cool? Oh, and by the way, I did bring it up. There's lysine 147, and there's the arginine 246 right there. So these two different studies find antibodies that like to bind the same place. The other thing is, check this out. SARS-CoV-2 patient, they made that purple antibody binds the MERS spike. So we finally found some cross-reactivity where we have an antibody against MERS. And actually, by the way, they also have antibodies against the beta common colds. OC43 and HKU1, those are the common colds that are more closely related. And they can see all sorts of antibodies binding in these. So the thing is, they looked at it both ways. And they tried to say, if there's somebody who had common colds before, basically general population, will they make antibodies against SARS-CoV-2? And the answer for that, unfortunately, is no. If you look at this, you have pre-pandemic sera, none of these bind SARS-CoV-2. But the reverse question, if you have SARS-CoV-2, do you bind other coronaviruses? And the answer is yes. You have binding, you see how you have numbers for SARS-CoV-2, obviously, but also SARS and MERS. So the good news is, probably if you've been infected with SARS-2, you probably have antibodies that you can make against SARS-1. And you might even have antibodies you can make against MERS. So the next time I go to Twitter and I see a bunch of people freaking out about MERS possibly spreading, I'm going to say to myself, I at least have one paper that suggests that even if MERS starts to spread, it would take a lot of mutations for it to get around our immunity. So I want you to, pay attention to that and the direction in which it works. Now, this applies to only antibodies. What if there's another branch of the immune system for which common colds can give mild protection against SARS-CoV-2? I think that is true for T cells, but that's what we're going to have to talk about on Friday. So the very last thing is that this is a preprint, so this hasn't been peer reviewed yet. I think it will be peer reviewed. I see a few problems with it, but I think it will survive.
notice how this group, they basically took polyclonal antibodies plus a spike protein, and they did cryo-EM of it. They saw where the antibodies were, right? And you could see multiple antibodies. Well, they took that a step further in this paper, because they say, what if you don't know the sequence of the antibodies at all? Could you possibly tell what the antibody was, what the amino acids are in that antibody? Could you sequence the antibody from just the cryo-EM? And that's what they're proposing to do. They're proposing to not sequence the antibodies. It's hard to sequence the antibodies. It's easier to sequence B cells, but those aren't antibodies. And so that's what this paper is, and I do want to just mention that this is possible. I want you to see the biochemical principle. The idea is, if you take antibodies out of a creature, a mammal, and they bind to an antigen, then you have the complex, and cryo-EM can capture that complex. What they're doing is they're getting another piece of information that allows them to not just say, oh, there's an antibody in general binding here. That's easy. But allows them to say, hey, that antibody has a tyrosine here, a histidine there, an arginine there. It allows you to say what amino acids are actually there at the interface. And so again, this is the easy part. The easy part is seeing where the antibody binds, and that was in the last paper. That works really well. You can see for each of these things, you have the antibody there, the antibody there. Okay. But how in the world can you tell which amino acid is there? So the first thing they did is they proved this with a polyclonal serum, where they, actually I'm not sure this is a serum, this has been purified in some way. It's a polyclonal sample of some sort, where they knew the sequence, and then they found that they could sort of reconstruct. You can't tell for sure whether this is tryptophan or tyrosine, but you can tell it's big. And you can tell this one's small. And from that pattern, if you have just a little more information, you might be able to solve this puzzle based on limited information. The way they provide the limited information is they have an algorithm that looks at the density, and then it says, oh, this has to be either, this is flat and twisted. It has to be either a proline or a glycine. This one's big. It has to be hydrophobic, so it's going to be HY or F, and they say exactly what those are. Where's HYF? Right here. Well, it's histidine, tyrosine, or phenylalanine. And here they, so they basically say this one must be an SSC, which means small side chain. So it could be one of these 5. So you don't know exactly which one it is, but you've narrowed down the possibilities. And what they do is they sequence the B cells, which have a rudimentary form of the antibodies. Remember, these are all coming from gene sequences in the B cells. So you get the repertoire of B cell sequences to know what can fit into this, and you match them up. And you eventually find a B cell sequence that uniquely fits even the vague sort of lumpy sort of residue electron density that you see there. And then you can figure out, okay, here there's a tyrosine, here there's a proline. And because I get the sequence from the B cell, and only that sequence will fit this pattern, even though the pattern's not exact. I hope that makes some sort of sense. I might ask you to choose to explain that because I think it's a really cool biochemical trick, and I think it's a really good review of what amino acids are. I told you they were important. So, that's it. Basically, we are beginning to see atomic detail of how antibodies are binding, and to me, this excites me because I've always been frustrated with not being able to see what an antibody actually is if it's not monoclonal. Most of our antibodies are complex and they're serum-based. We have dozens of different kinds. And so this is how we can actually know amino acids from a complex sample like that. And so I might have to take back everything bad I ever said about cryo-EM. It looks like cryo-EM is really the only technique that could be doing this for us. And I think it's pretty amazing.